Hey, boys and girls, weather report for June 15th, 2022. Why am I doing this?
That is the question, isn't it? Why?
Yeah. Anyway, Cell lays an egg. So Cell he went through a bunch of papers last night very rapidly because his computer was.
That should answer Professor Icarus's question in the side chat.
Oh, why? Why Cell?
No, yeah. He's not about, yeah.
Yeah, well, Cell stream yesterday. Yeah. Anyway.
I love Cell.
I thought this was amusing. There's a little part here, which we shall play.
Loadful, guys. That was not easy reading. All right, so let me try to uncoil a little bit of this. And I'm appalled that some of my detractors, who shall not be named, have been insisting that we're discovering more junk every day. And I'm like, what have you been?
What if I said that? Did I say that?
I think you may be straw manning you.
Maybe.
Been smoking, and in the case of one of my detractors, he smoked a lot, drank a lot, did a lot of LSD. So
really? Yeah. That again?
Okay.
Really?
Okay. I am not going to say that Cell burned his brain out chasing women and reading the Bible. I'm not going to say that on this channel, because if I said that, that would be like an ad hominem. So I am not going to say that Cell burned his brain out chasing women and reading the Bible. But anyway, let's see what he has.
Me to say what have you been smoking kind of really does hit home, because we have papers like this. So let me see if I could show a picture. Maybe a picture's worth 1,000 words where all this is. So, these RNAs, the junk DNA has a lot of repetitive elements. They're not exact repeats, though. And I've often said that what's important is it has a biophysical property.
Okay, now here's something where he is right. And he probably doesn't even know he's right, but he's right about this. Now we got to listen to this.
That means it changes its density. It changes the way it connects to things. It changes the physical property. So if you have like a stick, versus like a paper clip, you can see that they have different properties. Like the stick is very brittle. With the paper clip, you can bend it. And then you have, like if you have like silverware, each of the, certain silverware just bends differently, and you can, all sorts of things just have different physical properties.
You guys bend your silverware? I try not to.
My mama used to yell at me for doing that.
Yeah.
And I said the sequence affects the property. The sequence affects the physical property. So these things will coil up, glob up, take on different shapes, and then they kind of, when they're floating around inside the cell, they're going to take different positions. So
that's true.
We say to condense. So in the cell, they'll blob together in different positions inside the cell based on the sequence, especially inside the nucleus. And this creates how things are organized. It actually builds kind of the buildings and the compartments where everything works. This is really, really, Janet's here. This affects how the nucleus is able to organize all its cellular factories. You see, people have said, well, that RNA just kind of disappears. It's not done for anything. So ENCODE was saying a lot of this is transcribed. Now we know how.
Okay, so ENCODE has modified the position, and a lot of the stuff that ENCODE found was like very, very low amounts were transcribed, right? And we know there's a lot of transcription going on, a lot of spurious transcription of small RNAs.
Yeah.
And we have also known for many years about these biomolecular condensates, and we've known about the effects of microRNAs and small RNAs on all parts of the cell. And now if you have a cell and in your body somewhere, and it's mutated somewhat from when you were born, right? Every little part, every little molecule in that cell is going to have some overall effect on the cell, but usually it's vanishingly small. And if it is not vanishingly small, that cell will die through apoptosis, right? So, I mean, there's all of these little tiny effects going on, but that isn't what the paper he's about to bring up is talking about. They're talking about things where there's like RNAs that we know are transcribed and are useful. And he's extrapolating that to all RNA is useful, right? So these biomolecular condensates, I mean, if every one of them was doing something, we would have a real problem, right? I mean, it's like but so he was right in that everything, like that small effect is going to have some vanishingly small effect on fitness, and that's how evolution works. Because, there is all of this stuff that has no effect right now that could at some point lock into a pathway and become some sort of future beneficial mutation. So
Right.
he's talking about how evolution works. And where did we let's see, we got about 12 minutes to get to the part that we were looking at. Should we run a little bit here? Well, I've got some timestamps, so I don't.
I'm gonna
Whatever
see.
you want to do.
Oh, this one. Okay, this shows how excited he gets when he banned Mitchell.
Here, and someone will give me just a moment here. I need to take.
Somebody is saying something he said.
All right. Goodbye. Took care of someone here. Mitchell's gone from the channel.
Look how excited he is. I got to start banning people again.
I feel like I have a
come on,
modicum
Danny.
of power in this world. Oh my
I want
goodness.
Danny back. Bring Danny back so I can ban him some more. That's fun.
I recommend you go. There's some nice channels you can go to, like Speed of Sound's channel. So let me do next
He
one.
almost did the exact same thing to me when he banned me.
Yeah. And he's claiming that I don't have any comeback about what he says. Well, let's see. So let's see here, 3348. I don't know.
Well,
Talking about these are
first,
really
I believe
devastating.
he strawmans your comebacks because earlier
Yeah.
on he misrepresented or just
Yeah, I'm claiming
just
there's
made
more
up
and more,
something that
more and more junk DNA being found every day. Okay. Maybe he's talking about my, when I downloaded all of the signs, all of the lines, and all of the ERVs, and I've got like, I don't know, it's not terabytes, but it's a lot of data, and it looks like a lot of junk to me.
That's a lot of data.
By the way, I put a link in the private chat to that article referenced by Digital Hammurabi.
You found it?
Yeah,
Okay.
it's called New Discoveries in Iraq, Upend Story of Mesopotamia. And so I haven't read it yet. It looks interesting. And they're just adjusting some time ranges of, I think, when the Sumerians started irrigating, I think.
Ooh,
Right.
nice.
But I'll have to read the full thing.
And we have to go back to the beginning of this where he was talking, that paper by, let's see, who did that paper? I can't remember. The paper about the yeast and the synonymous and non-synonymous mutations, that synonymous mutations had an effect.
Right.
He's got that in the beginning of this, and I think we're going to have to address that. Because I went back and I found papers all the way back to, I think, 2006 talking about that same thing. In yeast.
Timestamps.
I linked most of the papers in the chat.
About the part where he talks about synonymous mutations?
I do. Yeah.
Okay, good. Because I'm interested to see what exactly he says there.
Well, we're dealing with, biomolecular condensates. We'll go through that.
Talking about these are really devastating papers for evolutionary biology. And thank you for subscribing to my academic channel. These developments are devastating. So if you caught it late, you might want to go to the
Devastating.
To the earlier part of this. So let's go here. This is another paper, Modulation of Phase Separation by RNA, a glimpse of N6-methyladenosine modification. We're not going to go through the whole paper. Phase separation.
Okay, he's going to read the abstract, right? And the abstract, they talk about liquid-liquid phase transitions and biomolecular condensates. So a biomolecular condensate is, imagine if you drop some oil and water, that the oil forms little droplets in the water. Okay, that's basically what happens in the cell. There are areas of the cell that form these little condensates. And they're kind of like little mini organelles in the cell. They'd be like not really a nucleus because they don't have a membrane around them. They're just that these this collection of RNAs and proteins happens to condense up into like a little droplet. And those things are important, and we've been talking about them for 10 years, but they're important in developmental biology and in a lot of your metabolism, or a lot of things in metabolism depend on concentrating things, various proteins together, and you'll end up with some sort of reaction. And they call it a biomolecular condensate, but you can just think, it's just a drop of oil in the water. I mean, and liquid phase transitions, basically that's what it is. Your salad dressing is a liquid phase transition, going from, watery stuff to the oily stuff, right? So, I mean, don't get caught up in the technical jargon.
Yeah.
Makes you sound smarter than you are. And this particular paper is talking about one particular modification, which is where we put like a methyl group, which is from methane. We attach it to one of the nucleic acids, and it behaves a little differently. And it's important in how these RNAs hook up to proteins and create these little globules. So that's all this is about. And I'll show you another video. And I've linked a whole lot of papers and a couple of good videos in my description. One of them is from iBiology, which we'll go over after we're done with cell here, or after we're done with the first phase of cell, and we'll go to that.
The first phase.
Yeah, so I'm going to skip the part where he reads the abstract and then he shows this illustration, which is, and this paper is, I linked the name and the author and the year, and if you pop that into Google, you'll find the paper if you're interested. And it is a very dense paper, but it is readable. And it doesn't say a damn thing about junk DNA in it. It doesn't say a damn thing about 100% functionality. It's simply talking about this mechanism. Okay.
It's powerful. So this is very good proof. This is excellent proof that all this repetitive junk DNA actually has a very important, can be modified. There's a few papers.
Okay. You want to comment on that? Okay.
I was just saying, like, he's using the term proof here, kind of exaggerate that a little bit. I'm just pointing that out here.
Yep. Yeah, this is very good proof, excellent proof that all of this RNA is being used. No,
I don't think that's the right terminology for it, Sonny boy.
Yeah, okay, the paper is right here. Can you see the paper? Yes.
Yep.
Well, PDFS, let's see. Okay. Yeah. So that is the paper that he's talking about. And this paper has nothing about all RNA is functional. Nothing. I mean, it's all about this one particular reaction, And here's all the clever little illustrations, and they are wonderful. And it is a wonderful process, and it's a beautiful thing, but it's a hell of a long paper, and he is probably going to read it, and him and Emery are going to go, oh, there, look, evolution is dead. I don't know what the hell. That's what I mean by him laying an egg. I think in a way he didn't understand the paper, so he may have thought that it was implying something that it wasn't. I mean, the RNAs that go into whatever they're talking about here, I don't think it constitutes all of the RNAs that ENCODE found. I think probably it's a couple of them, right?
Yeah, because there are lots of different types of RNAs.
Well, yeah, and the other thing is that because of this methylation process, you don't necessarily have to have a specific RNA to do this, right? It just, you have to have like a consensus sequence, and it'll work, and it'll create this condensate. And that's all they're talking about. So molecules are
like there's a word for that. I think it's called a hasty generalization fallacy.
Yeah.
I haven't memorized my fallacies, but I distinctly remember that one.
I like the sound.
I think that's a great example of one. You're right. Or you could just call it, he's wrong.
Yeah. Or it might be, yeah, I think it's either that or a fallacy of composition, because he's basically saying, oh wait, no, it's not a fallacy of composition. Yeah, hasty generalization, because again, and this is kind of the common trope with junk DNA overall with creationists, is that they'll find this one section or this one example that is functional or does something that happens to do something which again isn't necessarily indicative of biological function for various reasons. But they'll find something that does something or does a particular function, and they'll extrapolate that example to, oh, the entire genome must do something, which doesn't follow.
Another trick they like to use, this is a 2021 paper, they like to say, oh, science just discovered now that RNA has some effect on, whatever. But let me just pop something into Google Scholar here. Biomolecular.
So science is more about refining what we already knew, so to speak. Like a discovery is a good part of it, but it's not like, oh, we just discovered this brand new thing out of the blue. Like kind of what Newton said about standing on the shoulders of giants. New discoveries are built on previous discoveries most of the time.
That's very true. Yeah, especially science. Now, one question about the articles you list in the description, from the different papers. Speed, are you there?
Yeah, I am.
Did you mean those to be links or just
paper names?
Just paper names. So they're not okay, because they're definitely not linking.
It's much easier for me. Put them into Google and you'll find the paper.
Yeah. Google or even Google Scholar.
Okay, the condensates within the cell may improve our understanding of the dual roles of endogenous Nuke 98, 2010. And 2005, the fine structure and role of nuclei. You type it into Google Scholar. Let's see, where else? From atoms to single biomolecules. So that's a Bose condensate. So that's something else. But anyway, so this has been around for a while. So they've known about the nucleolus, which is a little weird structure inside the nucleus, is a biomolecular condensate. It doesn't have a membrane around it, but it groups together all this stuff that creates tRNAs and RNAs and all of that. And so, this isn't like breaking news, is what I'm trying to get at. It's not something we just discovered and all of a sudden evolution has returned. Right.
So do we need to cancel the investigative reporter we just sent out?
Yes.
Okay. Well, I'll
evolution
call
is
turn the van around.
Yeah.
Evolution has died so many times this week I've lost count.
I know. John Maddox killed it with what did he kill it with?
He said so many amino acid changes it becomes thermodynamically unstable. What the heck was he talking about?
I know. He said 3 to 15 amino acid changes and the protein becomes thermodynamically unstable. And I tried to find, it's like, John, where did you read that? What are you? Because I'm pretty sure that it's one of those Emily Latella moments where whatever he read had nothing to do with that. I mean, it's like, for God's sake, that's
right.
So you heard the same thing, or did we discuss this? Did we discuss this?
Well, you sent me the email, but yeah, I did see that part on I think his video yesterday.
Yeah, I mean, it just
I know what you're talking about.
What in the hell are you talking about, John? It's like, God, what these people. Okay, let's listen to this a little bit.
It's powerful. So this is very good proof. This is excellent proof that all this repetitive junk DNA actually has a very important function. It can be modified. There's a few pages in Contested Bones where I covered RNA, the RNA, one class of RNAs, the ALUs, but now it seems like it's larger than that have what we call the epitranscriptome modifications, where the RNA itself undergoes.
Wait, Dannykins.
What about microRNAs and all of that, all the stuff we've
wait, did he just call ALUs RNAs?
I think he did. Yeah.
They're not RNAs, Sal. They're repetitive DNA elements.
Yeah.
So I don't know what he's talking about there.
Cat, you're in the way, Cat. Thor, we have to talk. Go away. Don't look at me like that. God damn these cats. Okay, let's see if I can find this other video. Let's see. Study breaking.
The iBiology one.
Oh, that's what I'm looking for. Yeah. Here we go. Okay.
So I guess we're done with the first phase, and then we'll go on to the other thing he said.
Yeah, I don't think he makes any point other than that he found a paper that talks about RNA, like one RNA, right? And it's like all of them are functional. They're all functional. It's the same thing he did with ALUs.
Right. That's quite a grandiose claim there. That's quite a sweeping claim there, Sal.
It's just devastating for evolution. In his defense, he pre-believes in this creation model. So every time he reads something, his mind goes to, like, he extrapolates, to some conclusion. And he doesn't, as far as I know, in the video last night, he didn't offer any proof at all. I mean, or even any evidence to back up his claim.
Right.
But for him, he believes in it. He's not lying. He really believes that that's proof.
Well, the fact that he just provided one single example of this and didn't provide any independent examples from, like, I don't know, like a different DNA element or a different kind of RNA or anything like that to corroborate what he said. He just kind of picked out this one little example and extrapolated from there.
Yep. Oh, my
Righto, you are there, Andrew.
Okay. Now, so on this video here, what I want you guys to notice is on the very bottom where it shows that car all in pieces, and then it shows it together on the right. That's the same thing that James Tour uses. The same model that he uses, and so this is Cliff Brangwynne. He is working on these biomolecular condensates, and he's a real scientist, not a creationist. And yeah, now he's on YouTube. Oh man, that's Stephanie.
Okay.
He's hitting hard there.
Yeah. Getting some facts now.
Yeah, I'm trying to make friends with Sal, so I don't want to say nasty things about him. So I'm not going to say that he burned his brains out reading the Bible. I'm not going to say that on this channel. Okay, let's hear what he says about
how
this.
that works in science that we're trying to tackle. Now, this analogy between the parts in biological, in man-made machines and the way in which they come together to build things like an automobile, and the way in which the building blocks of
Sorry, just pause for a sec.
Really, I would say an incomplete.
No, for any creationists watching who will inevitably take this out of context, he said analogy. Okay, just hitting that point home.
He's got more to say.
Analogy. It's a useful way to start to think about it, but it's probably a flawed analogy. And the reason for that is because in biological systems, it's much more complex and rich in terms of how the components come together to build these structures. This is a beautiful simulation of the building blocks of life, things like protein and RNA and DNA. The way in which they come together to form biological structures is not like individual parts snapping together in static structures like as we would see for an automobile, but really dynamic, wet, chaotic, squishy, and constantly fluctuating. And yet these molecules in this wet dynamic soup are still able to come together and give rise to coherent motion, things like the leading edge of this cell that's protruding out to migrate and ultimately giving rise to the migratory behavior in this classic movie of a neutrophil chasing a bacterium, which is important for the immune response in our bodies. There's higher order. organization. This is gastrulation in the frog Xenopus. So many cells coming together to form complex three-dimensional structures. And it really is amazing to think that this dynamic wet soup of molecules can give rise to these coherent behaviors.
That's kind of cool. The way, yeah, and he
kind of included Brownian motion in there without even mentioning it.
Yeah, a little bit, yeah.
Yep. He's got some other things here that I clipped that I thought we should listen to. So this is about soft matter physics, soft
we
body
and others
physics.
are taking inspiration from other fields in trying to address these questions, in particular from the field of soft matter physics, which was where I trained as a graduate student. Soft matter physics is a really rich area that deals with many different types of materials, but they're unified in that they are soft with respect to deformation. And usually we're thinking about mechanical perturbation. So if we try to push on the material, it's easily deformable. But it could also be materials that are sensitive to electrical fields or magnetic fields and so forth. And one of the favorite examples of soft matter that I like are foams. And foams really are remarkable because they're materials that are made up of something like 95% gas and 5% liquid, and yet they come together in a way that forms an elastic solid, albeit a soft one that's easily deformable, but it really is an elastic solid. So this is something that I think escapes us in our simple notions of different states of matter that one can take without chemical reactions, 95% gas and 5% liquid, and make a solid. And that is pretty interesting. So there's this idea of emergent behaviors when you get materials coming together that have these soft character. Polymeric.
Okay, yeah, that's what I wanted to.
That's cool.
Talk about these emergent behaviors and
emergence. Gotta love it in the universe. Big thing.
Yeah, no, this is cool. Now, C. elegans. Instead of saying Caenorhabditis elegans, we could always just say roundworm. We wouldn't sound as smart, but anyway, this is talking about the roundworm embryo.
Yeah, species of nematode.
One of the first systems that we started to study in this area that really got me interested in these structures was in the C. elegans embryo. So this is C. elegans is an important model system used in many thousands of labs around the world. It's a small worm. This is the embryo of C. elegans, which contains these membraneless organelles called P granules. And these P granules, you can see in green here throughout the cytoplasm, are RNA and protein-rich assemblies. They're implicated in specification of germ cells, so controlling the fate of the cells that emerge through cell divisions and growth of this embryo. And it's important to try to understand these structures because similar granules are found in essentially all animals. So we'd like to understand what these things are and how they assemble and how they're playing their functional roles in cells.
And this has to do with how the zygote separates and forms the head and tail of the worm, basically. That's what he's talking about. And it has to do with these biomolecular condensates.
Now, when I first started thinking about these structures, I became very interested in something that happens in the very early stages of embryogenesis, where you see in this image on the left, P granules are initially distributed throughout the embryo, but over the course of about 10 minutes through development, they end up being localized to the posterior of the embryo. Now, this is important because when the embryo divides in two, it's going to divide down the middle, and only the cell that is on the right that contains these P granules. That cell will contain P granules, and the other cell will not. And that's the progenitor germ cell. And so we asked a very simple.
And that's
what are these states?
Screwed up. Where was I? I was on 11, and that's one of the really important parts about. One of the really important things in embryogenesis and how embryos develop is that like stuff will get slid over to one end of the cell and then the cell divides.
Are you there?
Speed.
We no longer can hear you. the video is frozen.
What happened?
When I first started thinking,
Oh, now it's back.
That's
Am
good.
I back? Am I back?
Yeah, you were
Yep.
gone for a second. The last thing I heard you say was stuff would move over to one side and then it would split.
Oh, okay. StreamYard. Damn it. Okay.
StreamYard.
So, StreamYard sucks. So anyway, that's very important in embryogenesis, that things will move over to one side of a cell, then it will split. Now you will have two different cells, and those two different cells will start to communicate with each other. The one on the left will say, hey, I'm a lefty, and the one on the right will say, hey, I'm a righty, and they'll go back and forth, and they basically create a third kind of condition between them, and then they'll separate again, and the cells that are between them are now betweenies instead of lefties and righties, and they'll get a different characteristic, and eventually out will pop limbs and brains and organs, and I mean it's the same kind of thing over and over again. Oh no, I triggered Scott.
Yeah, you trigger me. It's an old trigger. We haven't done that in a while, but yep.
Oh yeah, that's true.
Okay. Anyway,
thinking about this, these structures, I became very interested in something that happens in the very early stages of embryogenesis, where, as you see in this image on the left, P granules are initially distributed throughout the embryo, but over the course of about 10 minutes through development, they end up being localized to the posterior of the embryo. Now, this is important because when the embryo divides in two, it's going to divide down the middle, and only the cell that is on the right that contains these P granules. That cell will contain P granules, and the other cell will not. And that's the progenitor germ cell. And so we asked a very simple question about this process. I'm a big fan of asking really the simplest possible question at the start of a research project. And in this case, we asked the question, how are these P granules segregated to one end of the embryo? What is the physical and chemical mechanism that can take place? And can we try to understand that process using concepts and techniques and approaches from physics, for example? Now, in the course of this development, the segregation of P granules to the posterior of the embryo, something really interesting happens that we started to see when we very carefully quantified the dynamics. What we found is that early in the process, these P granules are actually disassembling throughout.
now I'm playing this because he does a pretty good job of coming up with an analogy that explains these condensates. So pretty soon.
The embryo. So all of the P granules are just slowly shrinking in time, as you see in this plot of the intensity or size of these structures as a function of time. But as you go forward and the embryo starts to polarize and define a distinct anterior versus posterior end, what happens is that the P granules that are in the posterior, which are labeled in red, start to actually stabilize and even grow in time, whereas the P granules that are in the anterior half, labeled in blue, they continue to shrink and go away. In the end, what that leads to is an embryo where there are only P granules in the posterior but not in the anterior end. So the segregation process reflects a spatial and temporal control of the assembly of these structures. Now, in the course of these studies, we started to ask questions thinking about the material properties, the material physics of these kinds of structures. We asked, what are these as physical objects, these things that are growing and shrinking? What do they feel like? And can that give us some insights into the assembly and disassembly? And we did a very simple experiment, again, in the...
Okay, I think this is the part where he explains it. Yeah.
Liquid-liquid phase separation. One can think about how this works as being something like if we were to take a box of water vapor at a uniformly high temperature and then cool down the end of the box, what would happen is the water vapor would condense into these liquid droplets and it would drive a flux of material into the posterior. So we proposed...
Okay, so I'm probably going to get copyright strikes for playing any of this. I don't know. I don't know.
We should probably stop playing a little bit.
God, it's such an educational video.
Yeah, it is. But anyway, it's linked so you guys can watch it. But
very interesting stuff.
Description.
Yeah, he's explaining it real well.
Otangelo said something brilliant here. Membranes and membrane proteins are interdependent.
So are we talking about abiogenesis?
I don't know what he's talking about.
Is that what we're talking about? It seems like he's wanting to talk about abiogenesis, and we're just talking about embryos and things.
Yep.
So
This is what I signed up for.
Oh! Anyway, got that out of the way. Feckenol. Don't dig too far. Hell's down there.
It made me chuckle.
well, although Stephanie accused me of not thinking about the metaphor and thinking too literally, but if you dig down through the topsoil and you run out of soil, then you hit either bedrock or exotic terrain. And so I would say that soil is thoughts, the bedrock is the framework of your worldview, your framework of epistemology and ontology and things like that that help you form thoughts and make sense out of them.
Now, here's another good question. If you dig down to the center of the earth, do you find dinosaurs and whatnot?
Yeah.
No, probably
Hollow
not.
Earth theory vis-a-vis the Edgar Rice Burroughs.
Yeah.
But you
story.
do find those cool slabs. Gee,
Right. Those
they're so cold.
prove young Earth creation. Yeah.
Always pack your long handle underwear because when you hit a cold slab, you're probably going to want some insulation.
And the ocean's worth of water. Don't forget that too.
No hell. Otangelo
What does
says,
he think?
It's not quite Christmas yet. I'm not in the mood. No hell.
Okay,
no hell.
Okay,
No
from his
hell.
last comment, is Otangelo implying that hell is literally the center of the earth?
I don't know.
Is that what he's saying here?
Who knows?
All the lost are at the center of the earth. I don't know what that. Probably some shrouded turn bullshit. I don't know.
Maybe. I don't know.
Yeah.
Anyway, let's get back to Sal.
Back to Sal. Love Sal. Sal's a great guy. Voted the #1 creationist.
And on genetic, so I felt it was important to cover it and talk about it. Also to show that it's really very premature for evolutionary biologists to be using evolutionary theory to claim that most of DNA is junk DNA, and by way of extension, the RNAs are also junk. I really think that evolution.
This fucking junk DNA thing, it's not a dichotomy. Okay, it's not an either.
It isn't. No.
And like,
I know we already knew that RNAs did things. It's like I was saying earlier, like just because it has activity, for example, doesn't mean that's essential to the organism.
right. Well, they keep tricking us into defending junk DNA.
Yeah.
And we don't even
Into
want to.
tricking us into falling into their binary thinking,
Yeah.
which is kind of a dysfunctional thing that a lot of people with a lot of thought disorders seem to do.
I better speed Sal up a little bit here. He's running a little slow tonight or last night. Okay. Got him sped up now.
I think, sorry, one more thing. I think, like, I don't know who, I forget who, but someone provided this, and I think it's a somewhat silly, but it's a good analogy for junk DNA. Essentially, a trash can smells, so it does something. Does that mean that's functional?
Yeah. The other one I like
odor molecules. Like, does that mean it's functional though?
Calling
My
it
garbage
junk,
doesn't smell.
okay, calling it junk drawer DNA is more accurate because there's stuff in your junk drawers that someday you might use,
Right.
And they're taking up space, and they're doing things that,
I
Right.
use stuff in my junk drawer all the time.
But I
I
mean,
reach through there and I go, hey, I can do it with this.
Probably non-essential DNA would be a better term for it. But that's
And then, of course, you have
catchy.
water spraying everywhere because I used the wrong thing.
Yep.
But, anyway,
Okay.
we're both speaking two different lines of thought, I think.
Yeah.
Oh, as we
All right.
often.
So what does Sal have to say about mutations?
What is and what is not functional. just based on evolutionary theory, the priority should be given to medical researchers, cellular biologists, molecular biologists, etc. We're actually studying the operation of the cell. That takes so much more precedence. We're going to cover some of the
some of the data that's the cell.
Okay, so he said, now listen to this again.
Centers, but a lot of them are like, if it's there in the genome, it's there for a reason, and I don't see why that is problematic. Larry Moran and Joe Felsenstein and Dan Grauer have a problem with that attitude because, as Dan Grauer said, if ENCODE is right, that is, if ENCODE is right, then most of the genome is functional, then evolution is wrong. And
I feel like that's taken out of context.
Yeah, why would that?
How would that make evolution wrong? It would just even if evolution is valid, even though a particular fact is wrong. If for some reason they observe data and the data, when it's all put together, says, well, there's a lot of non-functional DNA out there. If it turns out it all has a function, well, that just means evolution works a little bit differently than
right. Exactly. It means that we need to invoke a different hypothesis about how evolution specifically works. It doesn't mean evolution as a whole is incorrect.
So once again, they're inviting us to join them in their dysfunctional binary thinking,
dichotomous thinking. What are we gonna say, Speed?
Am I out again? Damn it!
You cut out for a second.
Yeah, what is going on with StreamYard?
I don't know.
Did you pay your StreamYard bill?
What Grauer was saying in his paper was that if more than 25% of the DNA is absolutely essential, then life wouldn't be possible because of the somatic mutations that would arise. You would end up killing yourself if you needed every single thing. I mean, if you couldn't stand any mutation at all, basically DNA does not replicate well enough to support life, basically. I mean, for God's sake, it isn't that complicated. There's a lot of space there where things happen that do not kill you. That's neutral stuff, right? And you got to have that neutral stuff. Otherwise, we die. I don't know. What do you think of that, Andrew?
It sounds reasonable to me.
Let's go on here.
Bit in my talk.
But how would that make it?
Did he just cut out again?
Yep. Maybe try reloading speed.
Maybe Minnesota slowed down their internet.
StreamYard it's StreamYard that's banking on me because everything else is working fine. I wonder if I leave the studio and come back.
Might make a difference. Why not?
Let me try.
Maybe instead of echoing, it's just making every other thing you say not go through. Ooh, he's doing some buffering there, isn't he?
Yep.
So Andrew, this is interesting.
Difficulties. I am big.
You are big. You are bigger than me.
I may as well go to OBS and dump StreamYard because they are not performing. For $50 a month, I mean, goddamn. I've got the can you guys hear me?
I can hear you now, yeah.
I'm paranoid.
Yeah, you said you might as well dump StreamYard, but I would say keep StreamYard for a while and experiment with OBS and see which one does you better.
Make a gradual transition.
I could bring you guys all in with Zoom. It's just that it's going to be difficult when it's not as easy to bring multiple people into the room. But I don't know. I think I can figure it out. But anyway, that way I'd be going through OBS, and OBS is already running on my computer, so it's already taken up bandwidth. So I may as well just dump this piece of shit. It's like God. The only thing about StreamYard that I like is where the heck is it here? Oh, I can't find it. I can't find it. StreamYard Virtual, Logitech. Oh no, General. Shift. Oh no, I can't find it. That's horrible. Too bad.
Uh-oh.
Oh no, I can find it. Here's what I like about StreamYard. Here's why I pay $50 a month.
Testing the speakers on StreamYard. Sounds like they're working. Have a great show.
That's why I like it.
That's really
That's
cool.
worth $50 a month right there just to have that.
That's $50 a month, and that was done by some of their techs. They got together and did that little ditty for them. Yep, that's the only reason I'm paying them. Okay.
Tuesday, you could actually just take the Poisson distributions and little population genetics. You could see that that is actually the correct understanding. So we'll just go right to the papers, and this is going to be boring and over the head of a lot of people, but such is science. And
No, it ain't.
you can just stop reading again.
How are you calling science boring?
Yeah, he does that over and over again. He said, "Oh, this is so boring." It's
Well,
like,
now I'm just mad at you, Sal.
yeah. How nasty.
Science isn't boring.
I know. Okay, let's see. We got it. Okay, he goes to this first paper. This is, which one is this?
And of course, this is the news article, not the actual paper.
Oh, yeah, it's the Michigan news article. Yeah. And of course, they made it sound more. Okay, so
Hyperbole.
I went back and I found a few papers on this issue. This is a brand new issue, right? They just figured this out just recently, right?
Yeah, let's show the historical context and then see what Sal misrepresents it.
Yeah, so let me see here. Oh, Ian. Okay, I got to go to a different screen. Okay, so can you see that one?
Yep.
Okay, that's Plotkin, so that's from 2011. That's on the same subject. This is the Shen paper, I think, that he's showing, that he's going to talk about.
Right.
Here's another one from 2020 that they're talking about it. Here's Walsh from 2019. Here's Drummond from 2006, basically talking about that. And there's Zhao from 2014. Paper after paper after paper. What the hell? I mean, just go look. Okay, They've found this stuff, and they said they would, in order to conclusively prove their point, they would have to prove this for other animals other than yeast. And yes, they do, and they probably will find some, but I don't think in general they're going to find that all synonymous mutations are going to affect the human being. I can point out Biolek, who wrote a book called Biophysics. He's got a little line, which I put a tab on, and this is a book that Sel likes to show, because Sel and I both own a copy of this book. And in that book, he explains that only 25% of some protein he was studying is similar, the same as these other proteins, yet they have the same structure and seem to do the same thing. Only 25% sequence. This is non-synonymous mutations, right? This is 75% non-synonymous mutations, and the protein still folds the same way, still does the same thing.
Here we have Otangelo going on the same bandwagon again.
Right. Yep.
I wonder if Otangelo actually understands Sanford's position about genetic entropy, what it actually says.
I don't know.
Because this paper actually shows the opposite of what Sanford is claiming, that all these detrimental mutations are undetectable for selection, basically. Because they have fitness costs. Which, again, isn't the new paper confirms. Yeah, you don't understand Sanford's position, Otangelo. I'm sorry, you just don't.
Okay.
Limit that I call Mueller's limit.
Oh, are you going to show the other screen?
Oh, okay.
That'd be good.
YouTube back on.
If more than 1% of the genome is functional,
he's going to go ahead and read this.
It's bad news for evolution. We're way past
1%. Did you catch that?
Do this correctly.
Listen to this.
All right. So the first part, there is a limit that I call Mueller's limit, and I've covered it on this channel. If more than 1% of the genome is functional, it's bad news for evolution. We're way past 1%, but
1%. He calls it. Cell created his own little thing. He calls Mueller's limit. If more than 1% of the genome is functional, then evolution is dead. What?
More than 1% of the genome is protein coding genes.
I know.
And RNA coding genes, and that's
so
really
much more.
arbitrary.
Yeah, it's like, oh, come on now.
Almost like he kind of chose that on purpose to kind of make his position seem more tenable.
I don't know what he's talking about. Anyway, let's see.
Finding more function here and there. This one, even by the estimates of Dr. Dan, might account for about 0.5%. This is what they call silent genetic mutations. I won't cover it much today, but I'm just highlighting
the other paper.
This article is in the video description.
Yeah, let's see what it is.
And it says study, most silent genetic mutations are harmful, not neutral. A finding with broad implications.
Yeah, and like I said, there have been papers on this for the last 20 years talking about Ka over Ks, basically. And it affects how you do population genetics. And there's a whole bunch of other things that go into that calculation that would affect population genetics, like some mutations are more likely to happen, and you have to factor that in. And we've known about this for a hell of a long time. And this paper came out, and the creationists have seized upon it because, you know, they said something about yeast. I don't know. Anyway.
And interestingly, it doesn't affect phylogenomics that much. But like you said, it affects population genetics, certainly. But actually, there are ways to account for it using different models of sequence evolution in phylogenetic methods. So it affects some areas, but not all of them for sure.
Yeah, because we use that to figure out what's neutral and what is being selected. So it gives us some indication of what's being selected.
Right. Of course, it's usually the case that mutations usually have at least some effect, but what matters is it really a strong effect to kind of make it matter enough to whatever particular method you're looking at, basically.
Yeah.
Because there's a difference between it has some effect, but it's really weak, versus it has some effect, but it's really strong and detrimental to the organism.
Yeah, and the reasons for one would be ribosomal pausing, but they haven't found a whole lot of evidence on that.
Right.
For ribosomal pausing, it's something that happens in bacteria, and the other would be the availability of tRNAs.
Right.
So yeah, if there was, and one of the papers that I flashed talks about that, and that would have some small effect on how much of the protein got transcribed or translated, basically, not transcribed.
Yeah, because we know codon bias has been a thing for some time now.
Yeah. But
these synonymous mutations aren't completely silent.
Right, and that's kind of what they're claiming. That's what the creationists are claiming. That this is, new paper.
Yeah. Right. Yeah, we kind of already knew that, guys.
Yeah. Okay.
Anyway, continue.
This nearly worthless definition of fitness called Darwinian fitness. This is bad. This is bad.
There he goes again with his,
my thoughts exactly.
Darwinian fitness. Darwinian fitness is fitting an organism fitting to its environment. That's kind of what Darwin meant by it, right? But then population geneticists starting in about 1920 defined it as reproductive success, which is what it should be, because that does have an effect on future populations.
Right. Who redefined it that way?
What's that now?
Who described it as reproductive success?
From like 1920 on, when the population geneticists got into it, they started
a little bit before the modern synthesis became a thing.
That really is a very succinct way to clarify it.
Yeah, they were all mathematicians, and they were interested in population models, and that's why, and it works very well for that.
Right.
And there's no other way to do it.
Like, how can we model this? How can we calculate all these variables for these populations, basically?
Right. No one gives a shit if the organism is some engineering marvel, which is what Cell wants to make it. That's how he wants to define fitness. Like, it's an engineering marvel. And evolution is marching forward to better and better organisms. No.
Spoiler alert, it isn't.
No. We're probably all going to die. It's just like all of us. I mean, it's not necessarily marching forward as an evolution. My God, what does he keep straw manning us that way? I don't know.
The previous anecdotal non-neutral synonymous mutations turned out to be the tip of the iceberg, said lead author Zhukang Shen, a graduate research assistant in Zhang's lab. We also studied the mechanisms through which synonymous mutations affect fitness and found that at least one reason is that both synonymous and non-synonymous mutations alter gene expression, and the extent of the expression predicts fitness effect. Zhang said researchers knew beforehand based on anecdotal reports that some synonymous
comment on that real quick?
Yep.
Yeah, so notice how they said the what effect it had on gene expression roughly correlates with the fitness effect. And I just want to highlight that because in the actual paper, they note that when they kind of, I guess, alter how they measure fitness basically a little bit, the fitness effect based on these mutations was highly correlated with the expression level of the gene. So like more highly expressed genes tended to, these mutations, synonymous and non-synonymous, tended to be more harmful, because if you have it, if you need it to be expressed at a certain level, a really high level, for example, it's more likely that's going to kind of offset that a little bit. That's going to be detrimental to the organism. So their findings, at least to me, aren't terribly surprising in that respect.
Yeah, and if you had, let's see, if you had a strain of yeast that had these synonymous mutations and somehow it was a small population and it drifted so that it was fixed in that population still survived, even though its fitness was somewhat less.
Right.
It could split off into a new species and later on, figure that out, right? I mean, eventually new things would happen that would change its fitness again. So, I mean, it really is no big deal. I don't know.
I also want to note that in the paper they admit and granted when they were switching between environments, or testing the fitness effects, they admit that their resolution methods, basically how the uncertainty was kind of a bit higher, but basically they found, accounting for that the percentage of synonymous and non-synonymous mutations that were strongly harmful to the organism varied between these different circumstances, which kind of makes sense. I mean, mutations depend how the effects of a mutation, whether it's detrimental or beneficial, depend on the environment.
Yeah.
So they're not always, this category or this set of mutations is always 100% of the time going to be really bad for the organism. That's
right.
Not exactly what they showed.
If that was the case, the organisms would go extinct.
Right.
We wouldn't have any organisms, but yet life goes on. Okay, so where are we at here? So 1713, I don't know, I got something here.
Let me tell you a couple things why this is devastating. First of all, a lot of evolutionary theories that talk about, they have theories that talk about divergence and stuff, and synonymous and non-synonymous mutations. There's called the Ka-Ks ratio. It's been a while since I've touched this, but it was based on the assumption that synonymous mutations were neutral. So when you look at protein phylogenies, you're assuming that the synonymous mutations were neutral, and that's false.
No.
It also shows that the proteins are most likely optimized.
What?
This is indicative that if the DNA is optimized sequence specific for the organism, by way of extension, we can assume that the protein is optimized sequence specific for the organism. Yes, it is true that you have mutations and variability, and you're not going to affect.
Okay, can I comment on this real quick?
Yeah.
So I don't know if Sal understands this, but how highly expressed or the translation rate basically isn't exactly dependent or tied to the specific function of the protein. They're kind of different selection pressures acting on both of them.
Yep. Yes.
So let me just make that point for the audience real clear here. So expression of the protein isn't necessarily tied to the exact function of the protein. They can evolve in a sort of modular way, basically. So what Sal is saying here isn't exactly correct.
Yeah, this effect that they're talking about is going to affect the quantity of that protein that gets translated.
Right.
And so it's going to lessen it. It's not going to completely destroy it. It's going to lessen it, so it'll go down from 100% maybe to 80%. And it's possible that that particular strain of yeast would become less reproductively successful by some margin, right?
Right.
Not completely. I mean, it wouldn't kill it. So, and that has nothing to do with the proteins being fine-tuned for a particular organism.
Because like I said earlier, certain organisms need proteins to be expressed at a certain level to get the job done, basically.
Yeah.
For their particular circumstances.
Hey, I'm going to go ahead and put the link out. It's been about an hour.
Okay, go ahead. Okay, so let's see. Let's go on.
Total
real
function.
quick. Sorry.
Slightly affected.
Oh,
I
what?
was gonna go quick for a sec.
Hey, sorry, I just noticed your link.
Yeah, well, when I send you a link, you don't notice it. What?
Yeah.
I mean, you sent it. Okay. Look, here's the thing. Next time, send it to the other email so I actually get it, because I have to go to my other email.
Okay, here's the thing, Midnight.
Speed.
I mean,
playing
okay.
Okay, we're playing a video.
The
Wait,
functionality.
real quick. Sorry.
Real quick.
I mean, well, Immutable just made a comment. Do you want to respond to it? Or
Expression of the protein is usually tied
exactly
to
is he saying here?
the stability of the mRNA that encodes it.
Could you elaborate a little, Immutable? Please?
Yeah, all mRNA is going to be susceptible to breakdown. I mean, it's around for what, 48 hours or something like that. But yeah, I don't think.
Oh, is he talking about like how certain RNA sequences can like degrade the mRNA after a certain period of time, and that kind of regulates how much of it is expressed or translated?
I don't know. MicroRNAs have a big effect on
Yeah.
whether an mRNA.
Yeah, microRNAs.
Maybe, I'm not sure. Anyway, okay. We're moving on. Hi, John. Hi, John.
Hey, John.
Hey,
Hey, John.
where are you, Speed?
Where am I? In Minnesota.
Yeah, you're not on the screen. You're not on the screen anywhere.
He's behind Sal.
I could be on the screen. What do you want that for? Okay, you want some of that? You got some. Okay. Let's listen to this. Where are we at? We're at 18. I don't know. Let's jog ahead to see what else I got here.
It's going to read this.
Sure.
It's going to be technical. I'll try to explain it away. And what's the takeaway says? What's the skinny? The skinny is evolutionary biology is wrong. It has gotten even wronger in light of some of the papers I'm going to read. They're going to be technical. So for the technical audience out there, they'll appreciate what this has to say. Okay, so let me see if I can just kind of increase the size of this. So even 2 years ago, I was having I had some challenges in defending the genetic entropy hypothesis because we didn't have as much data. But now 2 years later, this is,
Okay, so he shows a paper here where the guy suggests that they use a tool, a particular tool, and he goes through this method, and the paper doesn't have anything else in it that pertains to what he's talking about at all. So I don't know why he brought this paper up. I'm not sure.
Seems to be a pattern at this point.
Yeah, it does seem to be a pattern. Okay, so I just wanted to do that part where he does this this thing.
It's going to read this. It's going to be technical. I'll try to explain it away. And what's the takeaway says? What's the skinny? The skinny is evolutionary biology is wrong. It has gotten even wronger.
Wrong. It's wronger. Every paper they find makes it wronger. Yeah. Okay, and then he goes to he would show, and then I'm not sure where he goes next here.
Someone is saying something.
Oh, okay, that's that one. And then he goes to.
I mean, so look. I mean, once something steps across the line and is considered wrong, can it be wronger? I mean, it's if it's, you know what I mean? Like, it's already wrong. So like, what is wronger?
Or wronger.
Really devastating papers for evolutionary biology. And thank you for subscribing to my academic channel. These is powerful. So this is very good proof.
Oh no. Okay, I think that's it. That's all the links I got from him.
Oh,
So,
okay.
yeah, got nothing else. Don't need nothing else.
Yep, they're
Anyway.
really going at this new paper showing synonymous mutations having a fitness effect, which, like you said, one, we already knew, and two, doesn't support genetic entropy at all.
And hasn't been concluded for every species out there. We found
Right,
it.
that too.
Okay, the last paper, Immutable, I think, let's see, it's in my description. I think that's by Regy or something like that, was it? Let me see here.
I'm hearing dogs whining.
Yeah, that's my little lady. She wants to go outside,
Oh, okay.
and I'm not going to let her outside because I got a missing plank in part of the fence.
Okay.
Okay, there's the name of the paper. If you type that into Google, you will find a link to the paper. Sequence-dependent phase separation of protein-polynucleotide mixes elucidated using molecular simulations. And he talks about, I think in the abstract, they talk about using a program. And it's an interesting paper, but I don't think it shows that we're wrongorist. No wrongorist.
Wonder where Otangelo went. He seemed to head out right after we start explaining the papers.
Oh, God, I miss him already. Don't you miss him?
Well, Ox keeps deleting his comments.
Oh, he did? Why?
Who deleted him?
I mean, there's a perfect reason as to why people don't really all like Otangelo anymore, and they always delete his comments. I don't know if you want to talk about that.
No.
Why?
Well, yeah.
Backstage.
The controversy where he didn't apologize properly and all that, that's one thing. The other thing is that he's a spammer, and I don't find what he has to say particularly relevant. So have not enjoyed him. Let's see, what else was I going to say? There was something that tied into...
Are you going to shoot it in the head already? Oh, no.
Yeah, we'll go on for a little bit here. We did put the link out there, didn't we, for people to come in and comment?
I don't think so.
Well, I'll put it out there again, and I'll pin it this time.
Yeah.
Oh, yeah, that's how I got in here. Scott put it in the chat.
Yeah.
Oh.
We have a good question from Nano earlier. Does Sal have a degree?
I mean, from what I know, he's got 2, 1.
Yeah, he's got like an applied physics degree. Or is it math? I don't know, but he spent some time learning some biology, and now he thinks he's a biologist.
Oh, he's totally qualified to talk about biology, evolution, abiogenesis, and all that good stuff. He's totally qualified. Yep, they're totally qualified.
He's got a BS in applied physics and in math, and then he did some additional coursework in biology at the national. And it wasn't. I don't know what kind of degree he got for that, but it wasn't much. Anyway, that's what he's got. So
I will give Sal credit where he's due, and that he is, I guess, on the attack end against Ken Hovind and all the shenanigans surrounding him. So I will give him props for that at least.
And the way Maddox treated him at the end of the stream yesterday was horrible. I mean, what the hell was Maddox doing? Why was he so pissed at Cell because he got something wrong about John saying DNA is the code, and John was saying DNA is the medium for the code?
Well, that's because that's Maddox's standard argument, and like if he's kind of picking at that even in the slightest, then he feels kind of.
Yeah, but I mean, all Cell did was basically say it wrong and forget what John had said.
Right. And
then John just landed on him and wouldn't leave him alone. And he apologized and retracted it like 5 times. And he kept pouncing on him.
Well,
I think to be about Hovind.
Yeah. Well, to be fair, and this may sound a little harsh, but there have been cases where people have apologized to Sal for what they've said, and he hasn't gone back on that.
Oh, yeah.
So I don't know, just noting that here a little bit.
somebody who cannot be named.
I was thinking he may be getting a taste of his own medicine a little bit. Just pointing that out there.
Have you ever met John? Even if he's having a normal conversation, he sounds pissed off. Like John, John, how's the weather today? Ah, goddamn atheist, this and that. Like, dude, I just asked you what the hell the weather is like.
Yeah, atheists make
relax.
it rain, or atheists don't make it rain. Just depends on what you want.
Yeah. So I don't know. We had such beautiful weather here. It went from like 79 down to 66 with a thunderstorm, but the 66 feels like just yummy. It's just perfect because of the humidity. Oh, and we're going to have 102 next week, Scott. You'll be happy to know.
Yeah, it's 91.
Well, that just means we'll have 110.
91.
91, and then it feels like 102.
Yeah.
Supposedly.
Yep.
It's only 96 today.
Oh.
Well, to be fair, 91 is better than 110.
I mean, sure. What about 310? Can you survive that?
No. But doesn't water boil at like 212 degrees or something?
Depends on the pressure.
Oh, that's true. Yeah. That's very true.
Yep.
Imagine being Wolverine and surviving that.
Being who?
So I guess we could talk about like. Oh, sorry, go ahead.
No, I was just saying Wolverine. You know, like never mind. Go on.
Or I guess we could talk about Erica's earlier video today about plate tectonics.
Yeah.
Geology.
I linked that in the chat. Also, let's see, what else did I link?
Because I know you were there, Scott.
Dr. Dan. Dr. Dan has a premiere tonight with the complete Nathaniel Jeanson conversation.
Yeah, I guess he's going to premiere that.
Gutsick Gibbon had plate tectonics, Preclude, Young Earth Creationism, Bite-Sized Bus. Physics Girl had a video, which I put out, and Sir Sick had a video.
What was the Physics Girl video?
It was on time crystals.
State of matter?
Yeah, time crystals, I think it was.
Oh, okay.
And then, Scott, you mentioned something, new discoveries in a rock upend story.
It's an article that Digital Hammurabi linked to or put out a link to. Basically, it's going over when we think Sumeria started becoming a civilization. It was tied to their use of irrigation, but apparently they've got evidence that that's not so. So anyway, if you like ancient Near Eastern studies, you can't get more ancient than ancient Sumeria.
Linked in the description, my friends, along with many, many other things. So.
Did anybody see the article that I saw it going around the past few days about what they're calling common usage by ancient man of what they're calling like a caveman Swiss Army knife, basically like a stone tool that had a few different uses as far as cutting and what have you. And
I've seen the article, but I haven't read it.
Yeah, it's just making the argument that there was contact between human beings over long distances and somehow proving that through these stone knives that everybody commonly carried.
Yeah, there was different trade routes and then how things got from one place to another.
Right, and then like the stone tool from this area had like the same feature as the stone tool from that area, and like that kind of thing, So.
Or going back to geology, has anyone ever talked to? Oh, is there an echo?
I don't hear an echo.
Am I hearing something? That's weird. Yeah, so have we ever talked about radiometric dating on this channel? I don't think we have, at least not recently, right?
Yeah, because it's something we would have to learn a whole lot about.
I know
Erica has talked about it on her channel, and Maddie with Science Side Up on that channel has done several episodes about radiometric dating.
Well, I got let's see, indisputable evidence against radiometric dating from Reddit. Let's see here. Let's see.
Indisputable.
This guy is indisputable.
Oh,
Okay,
our creation, of course.
Who's Young Earth creationist? Who is
no
that?
idea.
I don't know.
No, I don't think that's his name. I think that's just a description.
A subreddit?
Oh, puzzle
Okay.
head. Okay.
Okay,
puzzle.
so wait, what is this indisputable? Okay, I have seen too many incorrect explanations and arguments around the radiometric dating process. I want my fellow creationists to be able to defend their position.
Okay, so the first actual step of radiometric dating is attempting to guess the original composition. Now, the reason they do that is so they can apply the right technique, right? Because if, like, that guy that Sal Cordova talks about that took, what did he take, collagen from a dinosaur and he sent it in? He says right in his paper that he fooled them. When he sent in the sample for dating, he told them that it was from, I don't know, what the hell did he say? Anyway, he lied about where he got the sample and what he expected, right? Because depending on what you expect, you're going to end up with the possibility of such a tiny amount of one isotope or another that you're going to have to apply different techniques and different, you're going to have to take different steps in order to date it. Anyway, he's claiming that that's bogus because that's why because he's claiming that we tell them what date to come up with. But no, that's not what they're doing. They're telling them how to treat the sample. And that's why you're supposed to be honest with your dating lab. You're not supposed to lie to them like this one guy did.
Yeah, if you lie to somebody, you're just not. You're just a liar. I mean, it doesn't say anything about the dating method. It says you're a liar.
Well, you have some idea depending on where you got it and what you got and how you extracted the material and, how you've treated the sample, all of that. You should have some idea what the hell you're sending in. And you should give your lab as much information as they can so they can use the most accurate methods that they have available. Right.
Okay, I like this. Wait, they also, by their own standard, at this point are assuming that the rock is older than 100,000 years. No. As long as the date ends up where they like, they check this box off. If not, must have been too young to date. There are other reasons why some of these things are discounted and not because of the age they give. Like, for example, because for any tool you use, there's going to be a certain error range, and if it falls outside that error range, then the tool isn't useful anymore. And like carbon dating, for example, using a freshly killed seal or whatever example they used back in the day.
I got a good question. Why are we looking at Reddit? Not a lot of people take Reddit seriously. So what the fuck? What are we doing?
Why are you asking questions? That's my question.
So the question we have from Midnight is, what the fuck?
Yeah.
No, I was asking why we're using a source from Reddit.
Well, because somebody brought up radiocarbon dating, and I just happen to have this on my browser. So because I looked at.
I didn't bring up radiocarbon dating. I just brought up radiometric dating.
Radiometric dating. I am sorry, I misspoke. Edit to. People didn't like the link I posted. I don't know, and nobody liked what he posted. So let's see. And he does calculations, and okay, who cares? We don't care. Do we care? We don't care.
I care about you, though.
Yeah, I care about you too. So, I don't know. Shoot in the head. Head shoot.
Yeah, because you and Midnight have to get a room, don't you?
John, shut the fuck up, John. Anyway, okay. Anybody got anything else they would like to discuss?
I got nothing.
I'm tired.
I could probably think of something, but I feel like that just.
So whose stream are you gonna get on tonight and annoy, Andrew?
I guess depends. Does anyone know if any of the usual suspects are doing anything?
Well,
My usual suspects. You mean Gurr?
No,
Okay,
not
so-
for.
Maddox put a stream up that he was going to have like 3 nights ago, and then he didn't take it down. Then he started another stream yesterday, and he still had people waiting on that stream for him to start streaming. But he
Oh,
may
he didn't
start
do that.
streaming-
That late night stream. He didn't do it.
No, he never did it. No, never did it.
I mean, you've had streams scheduled before, and
I know,
they
I
never
did it too.
materialized.
Yeah, but then somebody told me about it, and I removed it. Okay, Professor Dave, I think, has gotten some pushback on his gender video, so
Of course.
I was going to check that out. I'm not sure what it is. Cancel this, whatever. Anyway, there's that. What else? What else is going on? I don't know what else is going on.
And if SFT has done anything recently?
Oh, he does stuff all the Kent Hovind.
Yeah.
I mean, Jesus Christ.
Oh yeah. I just want to note, when Sal was going off on Kent Hovind in that Maddox stream, SFT was going bananas in the side chat.
Oh, really? Oh, that.
Yeah, he was saying some mean stuff. Let's just put it that way.
Oh, he's the one that was saying the shit about Cell.
Yeah.
Yeah, Cell doesn't have much good to say about SFT anymore either.
That's true.
He talks about him as badly as he talks about me. The thing is, what he implies in his show is that I don't have reasonable comebacks to his stuff. I think I actually do, and I think I sort of dig into the science of it, don't you think?
Yeah.
Yeah. I mean, I don't understand why he thinks my criticisms are so superficial, because they're not. I dig right into the paper. So,
okay, Gurr is saying that was a fun time. I was in LPP's stream. Which stream are you referring to, Gurr?
Yesterday, I think. Yesterday.
Oh, yesterday? Oh.
Yeah, and Cell was there, and then John comes in and just tromps all over Cell. I don't know why he did that. It seemed like something else was going on behind the scenes. I'm not sure why he got so pissed.
Oh yeah, and I think I interacted with SFT the other day. Well, obviously, I don't know why I'd expect anything less, but he still thinks I haven't came back at his ERV challenges.
Oh my God!
Just amuses me. I don't know. Like I said, I didn't expect anything less.
Do you know what I think explains Maddox? There's a whole bunch of videos on YouTube, road rage videos, where the guy stops his car, slams his brakes in front of the semi-truck, gets out of his car, and go and challenges them to a fight. Okay?
Right.
I think that explains Maddox. I think he's one of those guys that stops his car in the middle of the freeway, gets out of his car, and tries to fight with somebody because they cut him off or they were following too close or whatever. But I think those are called Karens.
I mean, yeah, it does kind of describe him at least a little bit.
You two on the bottom there, John. Midnight, you got anything before we end it here?
Not really.
John Maddox is a grifter.
Hey,
Jesus.
I miss that. Thank you.
Don't call him Quantum John, for the love of God.
Quantum John.
Why not?
I don't know, because Leo gave him that nickname, and I don't like it.
Yeah.
Oh, it sounds catchy.
I mean,
Okay,
it
guys.
is, but no.
We're gonna end this.
Okay.
Yep. Goodbye, cruel world.
Bye.
Answer.